welcome to the another video covering the placenta and membranes. This video is dedicated to potential and multiple choice questions, and I am also try to clear your doubts side by side and how to assess the question to give the right answer. Now question number 1, the structure which gives rise to placenta. Okay, now remember that there is chorion frondosome which is invading the decidua basalis. This chorion frondosome and decidua basalis, they together make the placenta. What is chorion laeve? Chorion laeve is the portion of the trophoblastic invasion that was present around the whole uterine cavity towards the other uterine cavity, and it does not give rise to placenta, rather it forms the fetal membrane chorion. And decidua parietalis also, this is the portion of the decidua where implantation is not taking place. So the answer in this question is chorion frondosome and decidua basalis. So any of the two option, they are correct, but I have tried to give you this question to tell you that if option chorion frondosome is present, it is also true, and decidua basalis, it is also true, but both of them, they will not be present simultaneously. Now tertiary villi are formed by, in the previous video I told you in detail that the implantation is completed by 11th day of fertilization. Implantation, not the formation of tertiary villi. So option 1 is not correct. Now day 21st of fertilization, tertiary villi is formed. Yes, it is formed by 21st day of fertilization. Tertiary villi is the meaning that the villus, in between the villus, first there is a trophoblastic invasion, cord-like structures, it is called a primary villi. Then secondary villi, there is the mesodermal tissue, it enters it, that is the secondary villi. And the tertiary villi means that the blood vessels have also entered this cord-like structure. So this is the tertiary villi, and it is formed by 21st day of fertilization. Day 21st of LMP, no, this is not true. Day 21st of LMP, by that age even the implantation is not complete. And 5 weeks post LMP, you can say either 21st day or 3 weeks after fertilization, 21st day of fertilization, or 5 weeks post LMP. In this case, answer option 2 and 4, they both are correct. I have tried to show you that this can be asked in any of the language, so you should remember this. Now coming to the question, a primary gravida gave birth to 4 kg girl. Her placenta will weight. We have learned that the fetal to placental ratio, weight ratio is 6:1. So if it is 4000 gram fetus, then the placenta in the ratio, if it is 6:1, it comes around 650. So answer here is 650. They can ask you 3 kg, they can ask you 3.5 kg, they can ask you 2 kg. So you have to put the ratio, proportion, and you will get the answer. Remember, it is 6:1. After birth of baby, placenta separates through chorion frondosome. Chorion frondosome takes place in the formation of placenta, but the placenta does not separate through this. Decidua parietalis, it is the part of the decidua away from the placental activity. So this is also not true. Decidua capsularis and decidua spongiosum, these two decidua, they are formed at the site of implantation, but the placenta separates through decidua spongiosum. This is the important part. Intervillous space is in placenta is lined by, we know that intervillous space is barred by the maternal blood. It is lined by single layer of cubical epithelium, no. Cytotrophoblast, no. Syncytial trophoblast, yes. In my previous video, I have explained in detail that intervillous space is lined by syncytial trophoblast at all sides, and it is called the villous trophoblast. There are two types of trophoblastic tissue, villous trophoblast and extravillous. The villus which is present in the intervillus, which is lining the intervillus space on all sides, and this is syncytiotrophoblast. All of the following are true except there is no mixing of maternal and fetal blood in placenta. True, there is no mixing. They are in close proximity, but there is no direct mixing. Intervillus space is lined by syncytiotrophoblast on all sides. This is also true. In previous question I just told you. There is anastomosis of blood vessels in branching villi. This is a confusing thing, because in my previous video I told that there is no anastomosis of blood vessel in the branching villi. This is important. Although most of the blood vessels they get anastomosis, but in placenta, in villi, there is no anastomosis. So the third option is wrong. Let us also see the fourth option. Near term syncytiotrophoblast of terminal villi are converted into thin and thick zones. Yes, we have already discussed it. The thin zones, they are called alpha zones. It is the place of gas transfer, and thick zones, they are called beta zones. It is the place of hormone synthesis. So option 3 is wrong. This is not true. There is no anastomosis of blood vessels in branching villi. All constitute the structure of terminal villi except. Let us see what is the structure of terminal villi. Cytotrophoblast, yes, it is present. Outermost layer is the syncytiotrophoblast. Then after syncytiotrophoblast there is cytotrophoblast. Then is the basement membrane. Then is the stroma and blood vessels, and in stroma there is present Hofbauer cells. Yes, they are also present, and Rohrstri. Now let's come to Rohrstri. These are nothing but the fibrinoid degeneration which are present in the intervillus space. They are not present in the terminal villi, but in the intervillus space. They are present near the terminal villi, but they are present in the intervillus space. So option 3, that is Rohrstri, is wrong. All are true about uteroplacental circulation. Uteroplacental circulation means from maternal and placenta, from maternal side to placenta. There are 120 to 200 spiral arteries. They open into the intervillus space. This is true. The spiral arteries, they open into the intervillus space. Amount of blood in intervillus space is 450 ml. This is wrong. We all know that even though the maternal size is bigger than fetal size, but in placenta more amount of fetal blood is present than the maternal blood, and the maternal blood present in intervillus space at one particular time is only 150 ml, and the 350 ml around it is of fetal origin. So out of 500 ml capacity of placenta, 350 ml is of fetal origin and 150 ml is of maternal origin. So option 2 is not true. Now let's see third and fourth also. Maternal blood in intervillus space is replaced 3 to 4 times in a minute. This is true. The capacity is 150 ml and the total flow in a minute is around 500 ml. So it is replaced 3 to 4 times in a minute. And in case of fetal side, the fetal blood which is present in placenta, it is replaced totally once in a minute. Now fourth option, syncytiotrophoblast in intervillus space has fibrinolytic enzyme activity. In my previous video I told you, because there is somewhat stasis of maternal blood in intervillus space, still there to avoid coagulation, the syncytiotrophoblast which is lining the intervillus space on all sides. This is very important. I am telling you again and again that syncytiotrophoblast is lining the intervillus space on all sides. Syncytiotrophoblast is lining the intervillus space on all sides, because this is the only interface of, it is the only meeting point of fetal and maternal tissue. So this line is very important. So it has, this syncytiotrophoblast has some fibrinolytic enzyme activity and it avoids the coagulation there. Second gravida aborted at 17 weeks gestation. She did not require check curettage after abortion. This is due to in my previous video, 2 videos I have told you that about the Nita Books membrane. This question is related to that only. Now see, the options one by one. Proper development of chorion laeve occurs by this gestational age. There is no relation of chorion laeve or chorion with the which can have relation with the need of check curettage. Check curettage is done to remove any remnant, fetal remnant or placental remnant in the uterine cavity after the product of conception is cast out. But in this, after 17 weeks, it is not needed much. Why it is not needed? There is no role of chorion laeve in that. Okay, so option A is not correct. Cleavage of placenta occurs through decidua basilis. Number 1, you should know, this is the wrong option. You should know that cleavage of placenta occurs through decidua, not through basilis but spongiosum. And this second option is not correct. Now, Nitabuk membrane is developed by this gestational age. By around 16 weeks, this Nitabuk membrane is the nothing but the fibrinoid degeneration which is occurring at the extravillous trophoblastic tissue which is invading in the decidua basilis. So, in between, at the junction of this trophoblastic tissue and the decidua, there occurs fibrinoid degeneration which limits the further invasion of decidua by trophoblastic tissue. So, when this layer is fully developed, the placenta is fully developed sort of, and when the cleavage occurs, it occurs because of this Nitabuk membrane. There is no haphazard in trophoblastic invasion. It is completed by this age, and when the cleavage occurs, it occurs fully. So, this is the answer is correct. This is because of the Nitabuk membrane is developed by this gestational age. Presence of Rohr's striae causes complete cleavage of placenta. This is also confusing option. Rohr's striae, it is present in intervillous space. You just remember Rohr's striae, it is present in intervillous space and it is of no clinical significance. So, the answer to this question is third, Nitabuk membrane is developed by this gestational age. All are transported by simple diffusion across placenta except. So, as the placenta has got nutritive, excretory, respiratory function, and the transfer of molecules take place from maternal to fetal and fetal to maternal side. So, they are asking transported by simple diffusion except. So, 3 of them are by simple diffusion and 1 by not. So, let us see one by one. Water, yes, it is by simple diffusion. Sodium and other electrolytes, yes, they are also by simple diffusion. Urea, confusing part is urea and ascorbic acid. Now see, let's see one by one. Urea from fetal to maternal side is transported by simple diffusion. It is the excretory role of placenta. And ascorbic acid, what is ascorbic acid? It is vitamin C and it is a water soluble vitamin. Water soluble vitamins, they are transported by active transport and not simple diffusion. So, answer is ascorbic acid. Urea, creatinine, uric acid, they are transported across placenta from fetal to maternal side by simple diffusion. Remember this. Now, similar question, all are transported by active transport across placenta except. Now, they have given the options that active transport is taking place in all of the following except. So, let's see one by one. Creatinine. In the previous question, I already told you that creatinine is transported from fetal to maternal side by simple diffusion. So, option 1 is wrong. Amino acids, yes, they are transported from maternal to fetal side by active transport. Iron, phosphorus, calcium and water soluble vitamins, they are all transported from maternal to fetal side by active transport. So, answer is here in this question is creatinine. Now, coming to HLA class 1 MHC are expressed by villus trophoblast. I already told you in the previous video that villus trophoblast, they do not because villus trophoblast is in direct contact with the maternal blood. It does not express any of the MHC, neither class 1 HLA class 1 nor HLA class 2. Extravillus trophoblast, which is causing the trophoblastic invasion into the deciduoabasalis. In the previous video, I have explained you that this extravillous trophoblast, it expresses the HLA class 1 MHC, only class 1 and not class 2. So the option is 2, second one, extravillous trophoblast, is correct. Now both 1 and 2 and none of the above. So you should be aware that villous trophoblast, which is in direct contact with the maternal blood, to avoid rejection, there is no class 1 or class 2 HLA, no expression. And the extravillous trophoblast, which is causing the invasion, and it expresses HLA class 1, only HLA class 1. Now this is about the clinical colors of the amniotic fluid question regarding that. Golden color of amniotic fluid is seen in, it is seen in RH incompatibility, post maturity, fetal distress and IUFD. So RH incompatibility, yes, because of the presence of bilirubin. Post maturity, it is greenish yellow or saffron colored, and in fetal distress it is meconium stained, and IUFD, it is, you may get tobacco juice or dark colored amniotic fluid. So answer in this one is first, RH incompatibility. Now next question, all of the following are true about umbilical cord except. Let's see the options one by one. We have to find out the option which is not correct, which is not true. Now Wharton's jelly, we all know Wharton's jelly is present in umbilical cord. Always remember Wharton's jelly, umbilical cord connection. It is formed by mucoid degeneration of extraembryonic mesodermal cells. Yes, this is true. I have given you detail in the previous lecture. You may go and see that. The false notes are because of local accumulation of Wharton's jelly. This is also true. False notes are very common, don't confuse them, and true notes are very rare, and always know how to differentiate between false and true note. Average length is 50 cm. This is also correct. Vasa vasorum is present in both umbilical arteries. This is wrong option. Vasa, in the previous lecture, you please go through the theoretical detail before attempting this video of MCQ. Vasa vasorum is absent in the umbilical arteries and veins. If you like the video, please subscribe and share, and thank you for watching. Any queries, please write down in the comment box and I will reply you back. Thanks a lot for watching.